starting this initiative, Savio. This is Savio's baby. He started it. I think it's an excellent idea. And let me say this, that as I said, it's a new one on the market that we're launching. It's kind of series of lectures held each month. And I think it's a good idea to kind of introduce one single topic, focused one single topic, as opposed to Miriam's Latin American hub, which I will say something about later on. And I like the name as well, NoRCEL Sessions. And I'm going to finish as soon as quick as possible, because I know Antonia, and Antonia is a very important person. He should be talking more than I should be. So anyway, NoRCEL and Savio go a long way back, since 2014, and our friend Marco Jose introduced to us. Thank you for that, Marco. And then we have written 3 collaborative papers that's I and Savio and his colleagues. And the last item being that Savio has supported NoRCEL by traveling to everywhere, wherever he's asked to turn up. And he always does. He never lets us down. Thank you very much for that, Savio. And of course, Savio also so busy that he arranged the next session with Dr. Alan Templeton. And that would be taking place on the 4th and 14th of April 23, that's next month. And so let me do a bit of the introducing to NoRCEL. Those people who don't know what NoRCEL is, my name is Sohan Cheetah, and I'm the founder and chairman of NoRCEL Network Researchers on the Chemical Emergence of Life. And what's the essence of E in NoRCEL? There are 3 aspects here. First one being E stands for emergence. And what is emergence? Life's emergence from prebiotic chemistry to prokaryotes. Then we have evolution, that will be speciation due to transformation and transmutation of living entities, as exemplified by evolution of prokaryotes to eukaryotes. And then the last one being extinction. Of course, sooner or later, everything dies. In particular, we're interested in extinction because we're now imposing these on ourselves and the rest of the ecosystem. In particular, I'm talking about footprint. Everybody talks about carbon footprint, but we actually talk about water footprint as well. And this is the team. Please get to know Professor Elias Chasterdor is on, and he was the one who was guiding me. He is the Chief Executive Officer of NoRCEL. Miriam is also present. She is head of Latin American Hub. And of course, there's this gentleman with a beard. He's Savio, and you know him well. You probably know more than I do. But anyway, he's such a nice guy. And then there is Oleg, he's the vice president. Martin is the president. Then we have Mukesh being the team member, international team member. Then there is Aya. She's the PhD student where NoRCEL is helping her to attain a PhD. And then there is Kathy McGrath. She's the administrator, and she's manning the Zoom today. Thank you for that, Kathy. NoRCEL Science Initiative, Astroscience Initiative, as I said, Latin American Hub. We're promulgating science in Latin America, and this year in November, it will be our second forum that we're going to talk about. This is different from, as I said, from Savio's, in the sense that Savio's is more focused, and whereas Miriam will give flavor from Mexico about the sciences altogether. And then there is the ASIN, Astroscience Exploration Network. This is particularly active in Africa. So we are actually on the verge of now signing a memorandum of understanding with the 3 universities in Nigeria to promulgate education in astroscience. Currently, I'm writing a curriculum for that. And then these are just the dates to remind you. The Latin American Hub date is going to be 11th of November 2023, and then ASIN will be held in Africa. And if anybody interested, you have to let me know, but the date is 1st of June and the 2nd of June this year. But I will give you further information on that very soon. And then there is this Blue Earth project. This is to do with the Earth's issue. As everybody talks about it, there seems to be the case that no one can do anything about it. I think our approach is different. We ask difficult questions. So 2022 question we asked was, is humanity settling its own fate on ecological survival? And this year, 2023, it was, is it time for planet B? Next year, it's going to be a human madness. And this particular forum always takes place in the month of January. So it's going to be on the 27th of January, 2024. But I won't let you off that easily. Let me just play you this 30-second video. Okay, this is my last slide anyway. So look us up and get to know your friendly network. We are always welcoming new members. I hope you'll be able to join. We've got a lot going on. And so that I pointed out at least 3 networks here, the Latin American Hub, ASIN, and Blue Earth Project, and now Sessions. And I like that, actually. I think Xavier is some sort of Superman who could think of something odd like this. All right, thank you very much, everyone. I'll unshare now and give away to our friend Antonio. Thanks.
Hi, Soha. Thank you very much, Soha. So, in this first session, it's a big pleasure to me introduce Dr. Antonio Lascano at Universidade Nacional Autónoma de México. Antonio is, in my opinion, one of the most important research in knowledge of life, and has been developing an extraordinary work in scientific dissemination in Latin America. And Antonio is type of research that no need introduction. So, Antonio, thank you very much for accepting my invitation. Please, can you share your screen? Please go ahead.
Thank you, Savio. Are you actually seeing the screen right now or not?
No.
Okay, then let me go back to. Now you can, I assume.
Yes.
Good, lovely. Okay, well, let me start by thanking Savio, who is a very, very dear friend and colleague, for the invitation to talk to you. And since I'm fully aware that all of you know the chemistry behind the Miller experiment, the significance, and so on, I decided to look at a number of issues that are normally not discussed, namely the historical background of the experiment, some of the impact that it had and in science, some of the issues that Stanley had to face when the experiment was about to be published. And finally, I will see how relevant the Miller experiment is to the issue of the origins of life, as we understand it now. I think it is. And some of the debates that are actually taking place in our field. So the title of the lecture is Prebiotic Chemistry. You all know what this means, and the Miller experiment, a retrospective analysis. And let me start by saying that all of us are fully aware that the experiment was done in the context of the so-called Oparin heterotrophic theory of origin of life, an issue that was proposed first in 1924 in the then Soviet Union, and then refined in an extraordinary book called Also the Origins of Life that was published in 1936 in Russian, and translated into English in 1938. Those were not easy times for people to devote themselves to issues like the origins of life. It was a time, of course, in which the Second World War had already started with the overthrow of the Spanish Republic by Franco forces. And I always recommend my students to do a very simple experiment. Go to the library and look at how wide, how thick are the volumes of scientific journals published during the times of the Second World War. And you see how thin they became because, of course, everybody was engaged in world efforts. But after the Second World War ended, there was a very intense reactivation of scientific activity, and the people reread once again the book on the origins of life by Alexander Ivanovich Oparin. The basic tenets of his proposal was that life is the outcome of a process. It's not an issue of spontaneous generation. There is a long evolution preceding the first living beings. For Oparin, the first living cells were fermentative bacteria, namely they were bacteria thriving on the organic compounds that were available. And for Oparin, this abiotic synthesis of organic compounds require, obviously, a reducing atmosphere. So once you have the synthesis and accumulation of organic compounds, then Oparin, who came from the Haeckel's tradition of life depending on protoplasm and not on a single molecule, proposed that quasarbates were actually an association of molecules that could interact, and from this interaction, actually the first metabolic processes evolved until the first fermentative bacteria actually emerged. Some people erroneously call this evolution in quasarbates of the organic compounds as a metabolic first theory. And this is a mistake because Oparin at the time, like many other scientists, that has to be said, was not aware of the role of nucleic acid. And for him, again, following the Haeckellian tradition, both genetics and metabolism resided in the protoplasm. And for him, quasarbates were a laboratory model of the ancient protoplasm. So this was the framework set up by the theory. And just to say something that I think is important, if you look at the theory, it actually says that the origin of life is the outcome of a process. Hence, if you discover water in another planet or organic compounds in comets or in meteorites, that is not proof that life is going to appear or has appeared in those other places. This is just one step in a long series of gradual evolution. And I think this is a very common misunderstanding that has to be addressed in one way or the other. Well, after the Second World War, and especially in the United States, a brilliant chemist, by all accounts, Harold Urey, had moved from the Manhattan Project into the Department of Chemistry at the University of Chicago. And he published in 1952 a classical paper that I strongly recommend you all to read. The paper was published in the Proceedings of the National Academy of Sciences. And with that paper, what Urey wanted to say was basically how was the chemistry of the primitive solar system, and in particular of the Earth. And he came out with a very elegant, very simple, but very elegant argument. If the Earth had formed as an outcome of the condensation of an interstellar nebula, quite clearly, the chemical composition of the primitive Earth must be something equivalent to the composition that we see in interstellar nebula. And of course, we know that in the universe, by far, the most common element is hydrogen, the simplest one. So Urey quite brilliantly calculated the likelihood that we had carbon as interacting with hydrogen as methane, nitrogen with hydrogen will give ammonia, oxygen with hydrogen will give water, sulfur with hydrogen will be sulfuric acid. And he published this, as I told you, in 1952. And then he gave a seminar at the Department of Chemistry at the University of Chicago. Now, attending that seminar were a number of professors, were a number of very brilliant members of the faculty and the students. Among them, and sitting in the front rows, was Carl Sagan. And sitting in the back, because he was always very shy, was Stanley Miller. And both of them told me independently the same story, that after Urey had finished doing his presentation, somebody got up and asked him, are you aware of the work of a Russian biochemist, Russian, not Soviet, keep that in mind, of a Russian biochemist that has proposed the same kind of atmosphere to explain the origins of life? And Urey said, no, I'm not, I haven't read those papers, but it will be interesting to see them. Well, eventually, Stanley Miller, whose first advisor as a graduate student at the University of Chicago had been Edward Teller, the father of the hydrogen bomb, but who had moved to Los Alamos to engage in the project of making the hydrogen bomb. And Stanley Miller was left without an advisor, and he went to see Professor Harold Urey and asked him to be his thesis advisor. And as far as we know, Stanley told me the story many times. He had told the story to several people. Urey at first rejected the idea of trying to do an experiment with these kind of gases, trying to simulate the conditions of the primitive Earth, but Stanley was insisting, and so at the end they came to a gentleman's agreement. Stanley will devote himself for 6 months trying to experimentally prove that this atmosphere was relevant for the synthesis of organic compounds, and if he had no success, then he will move to another project, which I think was a very wise recommendation. Well, Stanley very rapidly designed the experiment that you are very much aware of. There were 2 flasks connected by these tubes. There was boiling water in the small flask, and the water vapor came up and mixed with methane, ammonia, and hydrogen. And the mixture was subject for 1 week to the action of electric discharges. We normally speak of the Miller-Urey experiment, but in fact, there were several experiments performed by Stanley Miller. He designed the classical apparatus, which is shown here in the upper parts of this figure, but he also designed what I would refer to as a volcanic experiment, because he wanted to see if electric discharges acting on the gases coming out at high pressure from a volcano could actually lead to the synthesis of organic compounds. And he designed this very simple apparatus in which, with a sort of Pasteur pipette, the water came out, the water vapor came up at high pressures, and they simulated an experiment, a volcano. And he also had a silent spark experiment shown here that I will not refer to now. So I will speak basically about the classical experiment and the volcanic experiment. Now, Stanley Miller did the experiment, and many of us, many times, asked him what was the destiny of this original experiment. And Stanley always gave the same answer. It was very hard to clean it, so I threw it away. So the classical experiment ended up in the dustbin of the Department of Chemistry of the University of Chicago. But he left such extraordinary detailed instructions of how to perform the experiment that it has been repeated over and over again. And the pictures I'm going to show you now come from his friend, my friend, William Schoff, the famous paleontologist from the University of California at Los Angeles. So here you see the experiment at time 0. If we focus on the inside of the upper flask, you actually see the spark flying from one electrode to, not to the other electrode, but to the water that has accumulated on it. Imagine an electric discharge on the primitive Earth striking a piece of water in the ground. So this will be equivalent. And after 5 hours, William Schoff, repeating the experiment by Stanley, actually was able to see these extraordinary changes in the inside of the apparatus. Chemistry is a sensorial science. Of course, you have quantum chemistry, theoretical chemistry, and so on. But it is basically, it started as a sensorial science, and you can actually see how the composition of the inner part of the apparatus has changed. And it's a very dramatic change, actually, in only 5 hours. Well, Stanley actually took some samples of the liquid that had accumulated at the bottom of the lower flask, and he performed paper chromatography. He was able to detect aspartic acid, glycine, alpha-alanine, beta-alanine, a couple of alpha-amino and butyric acid, a couple of additional compounds. He was very, very fascinated, very excited by the results. He went to show them to Harold Urey, and Harold Urey told him right away, fine, write a paper and we will send it to Science, which was already a major journal at the time. So Stanley Miller did what a student normally does. He brought the paper, put his name in the first place, that of Harold Urey in the second author, gave the manuscript to Harold Urey. And when Urey had the paper in his hands, the first thing that he did was to erase his own name. And his argument was very simple. His argument was that the experiment had been designed, done, analyzed by Stanley, and he told him, you are the one that deserves the recognition of this work, not me, that I'm already famous, which I think is an extraordinary example of the high standards, ethical attitude that Urey had in many, many places, but in this particular case, it's quite obvious. So Stanley did so. He erased Harold Urey's name, and then he sent the paper to Science. And all of the documents that I'm going to show you now come from the archives of the University of California at San Diego. They are very generous people. They are very organized archives, and you can actually go through them without any problem. And so there you find the response from Science. On February 16, Stanley Miller received this small card saying, thank you for your communication on a production of amino acids under possible primitive Earth conditions. You will hear from us later regarding the possibility of its publication. A famous phrase that all of us fear whenever we receive this kind of answer from a journal. And so, but Stanley was quite confident of the results he had achieved, that of the analysis he had done. And then a couple of weeks later, he was actually reading the New York Times, and the Sunday issue of the New York Times, because you know it's actually quite thick. And then he read the following note, which I am transcribing. In Ohio State University laboratories, Dr. Waldman McNevin and his associates are recreating the Earth's condition 2 billion years ago, long before there was life on the planet. Of course, we know now that life is older, but still. One one purpose of the study that Dr. McNiven explains is to answer this scientific question. Did the extreme complexity of chemical compounds develop before life appeared, or was this a result of life processes? Some of the experiments done by McNiven and his associates led the researchers to the conclusion that natural heat, light, and electricity could bring about highly complicated chemical reactions before living organisms evolved. Artificial lighting was also used because there is evidence that lighting was quite continuous in the early days of the Earth. 100,000 volt spark was sent through an atmosphere containing methane, marsh gas, and water vapor, produced resinous solids too complex for analysis. So when Stanley saw this, he almost had a stroke because, of course, it was basically a description of what he had done. So he rapidly sent a note to Professor Urey. The note is here again from the archives at the UCSD. And he says, Dr. Urey, this was sent the next day, March 9, that I'm sending you a clipping that I saw. Let me transcribe it here. I am sending you a clipping that I saw in this Sunday New York Times. I am not sure what will be done now because their work is, in a sense, my thesis. So he felt that he had been scooped, which is something, of course, we all fear. And Urey was absolutely infuriated. So he very rapidly sent a telegram to Lucille McCormick, who was his secretary. He was away giving some lectures, and the telegram said, send Miller's paper to the Journal of the American Chemical Society, Urey, University of California. He was truly, truly quite upset. So Stanley Miller had to write to Science, retire the manuscript, and send it to JACS, to the Journal of the American Chemical Society. And then when this happened, the editor-in-chief of Science, Howard Meyerhoff, wrote a letter to Stanley saying, although your paper, A Production of Amino Acids Under Possible Prebiotic Air Conditions, was submitted to us by Harold Urey, developments of the past days prompt me to turn to you as the author of this paper to seek your advice and instructions. Dr. Urey sent your paper to us on February 10. It reached the editorial office on February 14, and on February 16, it was sent to a referee for an opinion. Well, what had happened was that the referee saw the paper, could not believe the outcome of the experiment, and then put it in a drawer and forgot about it. And in the meantime, what Urey had done was to give in a seminar in Ohio. He spoke about the experiment. He felt very proud of Stanley, very proud of the results. So he spoke about the experiments, and Lake McNevin very rapidly decided to repeat the experiments. But Meyerhoff actually wrote to Stanley, the manuscript will be held here until I receive your instruction. If you are willing to let it remain in our hands, arrangement will be made to use it as the lead article in Science. So then Stanley wrote again to Professor Urey, told him of this offer by Science, and then Harold Urey told him, okay, you can ask the people from JACS to send you back the paper and let us remain in Science. And Stanley had to send a telegram to the editor-in-chief of the Journal of the American Chemical Society saying, please return manuscript mailed March 13 by Harold Urey entitled Production of Amino Acid. A mistake was made in sending it to you. Regret this inconvenience. And Stanley felt, well, okay, perhaps they think I'm just a fool, but in any case, the paper was published on May 5th of 1953 in Science, and here you have the paper published very rapidly. Now, Stanley, keep in mind what I just told you. Some of you actually met him. Stanley was a very shy person. And the impact that the article had, not only in the scientific circles, but in the media, was quite extraordinary. And here you see a collage I made for some of the notes that appear in the newspapers, not only in scientific journals, but also in newspapers like Los Angeles Examiner, Los Angeles Times, the New York Times, and so on. And all of a sudden, Stanley saw himself in the spotlight. I mean, always being very timid. And all of a sudden, there was a tremendous amount of attention on him. And he felt that he was not able to cope with that public attention. And he said, what if I devote myself to some other thing, like, for instance, become an artist? And he decided to take paint lessons with his cousin, a wonderful lady that I had a chance to meet once. She was living in Seattle. And Stanley produced this single painting. Believe me, the photograph is much better than the original, who is in the hands of a Maria Morris, Stanley companion. And when his cousin saw the painting, she told him, Stanley, I think you should go back to science. So, okay, Stanley decided to forget what was not a very promising career in art, in visual arts, went back to science. And the question was a very simple one. If he wanted to graduate, and remember that he had done the experiment when he was only on the first and second semesters of his PhD studies, he had to understand what had happened between the original setup of the experiment and the final outcome with all these organic compounds. In other words, what Urey told him, you have to understand how you go from methane, ammonia, hydrogen, and water into amino acids, hydroxy acids, and urea. So Stanley started doing the kinetics, the dynamics, the intermediates of the experiment. And once again, if you go to his notebooks, which are also in the hands of the archives of the UCSD, here you see the watermarks of the come with the copies. They are very generous people. They let you examine everything. And keep in mind, we were talking of 1950, the early 1950s. The level of chemical resolution of the available techniques was by no far as fine as the ones we see today. But you see, he was working with 4 significant figures. So he was really very, very, very careful. So he was actually able to deduce that what we actually see is an experiment during which aldehydes are formed, hydrogen cyanide is formed. There were not original precursors in the experiment. You see the downfall in the concentration of ammonia, and you see here the curve of the amino acids. So for him, when he saw this, he could very, very rapidly recognize the so-called Strecker reaction that was described by Adolf Strecker in 1850, and that basically says that you have an aldehyde that reacts with ammonia, with hydrogen cyanide in the water, in the gaseous phase. This is a reversible reaction, and then hence you have the formation of aminonitriles, and this aminonitrile, this is a very reactive group, reacts with water hydrolysis and gives a carboxy group. So here you have a second part of the reaction, an irreversible reaction in which you are led to the synthesis of amino acids. And something equivalent actually occurs with the formation of hydroxy acids, which Stanley also had reported in the experiment. Well, with these results, eventually Stanley graduated in 1954 very rapidly. He did his thesis very, very rapidly, got his PhD, and then moved on first to Columbia University to do a PhD, postdoc, sorry, then to Caltech, and he started and eventually moved back to UCSD. He moved to UCSD, where he got a permanent position as professor of chemistry. And then Stanley actually kept working on the origins of life for many, many years. Once I was there doing a sabbatical, and Stanley came in very excited with a fax from the Museum of Natural History of South Kensington in London, and they were asking him for samples of the original experiment. They wanted to buy the samples of the original experiment and wanted to put up in an exhibition. Stanley was extremely proud. They were offering him a high price. He was quite proud by the fact that they wanted the original material. And I told him, Stanley, of course, you don't have any samples of the original experiment. And he told me, of course, I have them. So we went to his office and he pulled down a box, a cardboard box in such way. There were many, many little boxes like the ones you see here. And every little box contained samples of all the experiments that Stanley had done in 50 years of work. You see here the run of experiments, the precursors that were used, the page in the notebook in which they are described, and whether the paper was published or not. Perfectly organized. I was quite impressed by this, and then Stanley actually showed me the samples of the original experiment, which you can see here. I was quite moved by it. I asked him to take, if I could take a photograph. He accepted, no problem. And then I told him, Stanley, are you going to sell the samples to the museum, or what do you plan to do? They should be in very good hands. And he told me they will remain in good hands, and he gave it to me. He gave it to me, which was, of course, quite an extraordinary gift, and I keep it at home. And for instance, yesterday, I took it to my students in my undergraduate class so they can see actually the outcome of a very, very famous experiment. Well, a few years later, Jeff Beda, who is a very able geochemist, he's probably the major expert on the geochemistry of amino acids. He was the second student of Stanley Miller, a second PhD student of Stanley Miller. He and I were invited to Austin to give a seminar on the origins of life. And the night before, we were comparing our slides. And when I showed this to Jeff, he was quite excited, and he came up with the brilliant idea that we should analyze this with the modern techniques. So some part of the samples were sent back to La Jolla, and then we actually design a whole experimental procedure together with a number of friends, Jason Dworkin, Danny Glavin. You will see their names in a moment. And we rapidly realized that we had not only the samples from the classic apparatus that I just showed you, but also we had samples that Stanley had kept from the so-called volcanic apparatus. So very rapidly, the chemical analysis was done using a combination of chromatography and mass detection. And here you have the authors, Johnson, Clips, Dworkin, Glavin, myself, and Jeff Beda. In the upper chromatogram, what you see are the outcome of the original experiment. But below, you see how actually a much more abundant number of amino acids are formed, and in a greater concentration. Remember that the area under the peak corresponds to the concentration of the compounds. So we were always very surprised to see that Stanley decided not to publish the results of the volcanic experiments, concentrated only on the classical experiments that also made quite an impact, as you have already seen, as you already know. Well, we analyzed in detail the outcome of the volcanic experiment. Here you see the results. You see a number of amino acids, several that had never been detected before, like homoserine, 2-methylserine, beta-hydroxyl aspartic acid, and so on. And one point that I would like to underline is the following. Here you see that we were able to detect valine, isovaline, and norvaline, which are all isomers. The one that we see in living cells today is valine. We don't see any isovaline or norvaline. In other words, we have here 3 5-carbon amino acids. They are isomers, but the only one that you see is valine. And in fact, isovaline is much more abundant than the two others. And I think this is a reminder of something that we have to keep in mind. When we think about the primitive soup, one shouldn't think that exactly what was needed for the origin of life was formed. And we should not think that the most abundant components were the ones that were incorporated into living beings. Clearly, neither of those arguments hold for isovaline or norvaline, don't hold for valine. So clearly, there must have been a selection process that must have depended on the stability, on the efficiency of the synthesis, on the ability to not cause stereochemical issues and so on. So I think this is a good reminder of this. Well, the paper was published. We were all very happy. There was an extraordinary recognition for the carefully kept samples by Stanley, by the insight of Jeff Beda to do an analysis, and we were quite happy with it. And then several years afterwards, I remember very well, I was flying to Zurich, and then I got an email from a very dear friend, friend of several of you as well, Dave Dimmer, who told me, you should read quite rapidly the last novel of Dan Brown called Origins. I have to say that Dan Brown is not my favorite author, but he has written the Da Vinci Code and Angels and Demons and so on. And he actually had just published, as Dave Dimmer told me, a book called Origin. I got off the plane, went to the bookstore at the Zurich airport, brought the novel. I was not enticed by the novel. I have to say it's not my kind of literature. But as I went through the pages, I found something absolutely fascinating, which is this paragraph that I'm describing to you now, I'm showing to you now. Edmund is one of the characters, Exalt. However, as many of you know, the Miller-Urey experiment failed. It produced a few amino acids, but nothing else resembling, nothing even resembling life. Miller and Urey eventually abandoned their experiments. The religion and their experiments. The religious community breathed a sigh of relief, and the scientific community went back to the drawing board. He paused, and he paused, an amused glimmer in his eyes. That is until 2007, when there was an unexpected development. Edmund told the tale of how the forgotten Miller-Urey test tubes. Sorry, I actually cannot see. Let me stop for a second. Okay, now I can see everything. Edmund told the tale of how the forgotten Miller-Urey testing vials had been rediscovered in a closet at the University of California in San Diego after Miller's death. This is not true, of course. Miller's students had reanalyzed the samples using far more sensitive contemporary techniques, including liquid chromatography and mass spectrometry, and the results had been startling. Apparently, the original Miller-Urey experiment had produced many more amino acids, this is true, and complex compounds, this is also true, that Miller had even been able to measure at the time. The new analysis of the vials even identifies several important nucleobases, the building blocks of RNA and perhaps eventually DNA, which is absolutely not the case. But in any case, it was very amusing to see how an experiment was actually made it into a very popular, into the writings of a very popular novelist. And my hope is that eventually we will be made into a movie, and I will have the chance to walk in the red carpet in Hollywood. But in any case, this shows you how into popular culture, sometimes work on the origins of life can be incorporated. Now, so far, I have described you only issues that have to do with science, with the experiment, and with these actually projections into popular literature. But there was also the socio-political context, especially the young students, the younger scientists attending this lecture tend to forget or are not aware that the 1950s were a very, very complicated part of the history of the world in the middle of the Cold War. On the Soviet Union, you had the extraordinary repression against the critics of Stalin or his successors. And in the United States, you had the very terrible situation of McCarthyism. And in the middle of that Cold War, Stanley received an invitation in 1956 from Oparin, who told him that he was organized in August 57, a meeting on the origins of life, the first international meeting on the origins of life, and that he was being invited to present a paper. Stanley was fascinated with the idea of going there, but he was not quite sure that he should go. I mean, he could be accused of being a communist. And he wrote very rapidly to Harold Urey, asking him for advice. Again, this comes from the archives of UCSD. And although it's not very clear to see, let me transcribe what Urey answered. I do not know how to advise you. I think each of us must make his own mind about this. The nuclear scientists from the USA went some time ago, and if they let the nuclear scientists go in the United States without stigmatizing them, I should think that innocent people like us might also go, but one never knows what McCarthy will do in the future. It is a very sad situation. It was indeed a very sad situation. For instance, Linus Pauling had his passport removed by the U.S. State Department, so he would not travel. Stanley Miller wasn't sure that he should go, but then he decided to apply for money to actually make the trip. And here you have the letters also found in those archives that I mentioned to you, in which Stanley wrote to the National Science Foundation asking for support, for monetary support, and who answered him was the United States Atomic Energy Commission, the director of the Security Division, asking him that, okay, if you go there, you must report any significant changes in your itinerary. You cannot disclose any restricted data to which you have had access under a program of any authorized person, and to report to this office by mail or by telephone upon your return to the United States. And then he brought again now to the following the advice he had received from very high officials that had nothing to do with science. He wrote to the State Department, and he was in fact agreed not only to get the permit to go to the Soviet Union, but also to go and to get the support. Now, there's one thing, it shows you the kind of atmosphere there was in the United States, in the world at the time. But one thing that I never asked Stanley, and I'm very sorry about it, I did not become aware until later on, was the fact that Stanley's main intellectual and scientific influences were offering a Soviet science, very close to the Soviet establishment, a supporter of Lysenko, never a member of the Communist Party of the Soviet Union, but clearly someone with ties, unholy ties in the eyes of the McCarthy people. John D. Bernal, who was a brilliant scientist by all accounts, who was actually a member of the British Communist Party, and Harold Urey, who was never a militant in any kind of party, but who was a strong opposer of the Committee of Anti-American Activities led by McCarthy. So Stanley, being such a very shy person with no political experience, was actually publishing papers, writing under the intellectual and scientific influence of people that were not seen as probably the best people you could think of in political terms. But in any case, Stanley went to the Soviet Union. When he went back to the United States, he was actually visited by what the people used to call G-men, members, people from the government, who did an interrogation of him, a very simple one about what he had seen, what he had told, and so on. In any case, by then, the experiment had already become part of the classical experiments in science, of a key experiment in the origins of life. And now we speak many times, many of us, about the RNA world and so on. Well, the history behind the RNA world hypothesis, however you want to define this hypothesis, actually has to do with the development of organic chemistry. It clearly starts with the synthesis in the 19th century of urea, sugars, alanine, by chemists that had not in mind the origins of life at all. They were trying to understand the way plants actually do the autotrophic assimilation of carbon. But then Oparin, especially Oparin, but Haldane, Lippmann, and Harvey as well, turned towards these experiments. They were very, very keen on these results. And instead of arguing whether this was the mechanisms or not for the origin of the sugars, of the organic compounds that we see in plants, uses as examples of the processes that may have led to the formation of the primitive soup. This led to the proposal of the heterotrophic origins of life. The Miller experiment was clearly a key point, not only history of origins of life studies, not only in history of evolutionary biology, but in science in general. And then in 1961, Joan Oró, a very dear friend of Stanley Miller, Joan Oró, who's this year is the centennial of his birth. I just came from Barcelona from a meeting celebrating his 100th anniversary of birth, reported the synthesis of adenine, and eventually the origins of life evolved into the proposal of the RNA world concept, which of course has been contested as it should be by science. Well, let me see. So then now we can try to actually very critically look at whether the Miller-Urey type experiments are valid models of prebiotic synthesis. Well, first of all, and I'm sure you are very much aware of this, the primitive atmosphere has not been as reducing as originally assumed by Urey, by Oparin. It is CO2 and not methane may have been the dominant carbon species. This is an overall description of the primitive Earth because they might have been a local environment in which the reducing conditions were higher. Secondly, different organic compounds are formed under separate experimental conditions. They are not always truly compatible among them. For instance, if you analyze the result of the Miller experiment, you never find adenine, or you don't find purines or pyrimidines, you don't find sugars. Thirdly, if you want to have an RNA world, you need sugars. And the problem is that sugars easily polymerize and form tar-like mixtures. And the question is, how compatible are prebiotic synthesis and the RNA world hypothesis? Well, to address the first question, again, with in the group of Jeff Beida, and this was actually the last paper co-authored by Stanley Miller, we decided to see if by keeping constant the pH of the water solution and using an atmosphere that was neutral with CO2 instead of methane, with nitrogen instead of ammonia, with no free hydrogen, we could form amino acids. And indeed, we got a very good deal of amino acids. It was already published years ago in Origins of Life. But these kind of experiments are what I call, or many people call, one-pot prebiotic simulations. If you look at the Miller experiment, you put all the necessary precursors, the source of energy, and it's in one single cazuelita and one single pot, you actually find the different compounds. The problem is that, as I already mentioned, the different organic compounds are formed under separate experimental conditions. And it's not evident how this could have led to ribonucleotides or nucleotides or RNA in the primitive Earth. Let me give you an example. If you want to have the abiotic synthesis of ribonucleotides, there are some arguments about the sources of phosphorus, but we can come out with good solutions. We have a very good synthesis of sugars, especially ribose and deoxyribose. And of course, we can form without any problem. Much more difficult than with purines, but we can form the pyrimidines. But if we want to form cytidine, clearly we need to, in some point, we need to form a bond between the pyrimidine and the sugar, and we need to phosphorylate this sugar with the phosphate. And these reactions are almost impossible. The difficulties in synthesizing this bond are really, truly beyond description. And because of this, and with other problems, John Sutherland came out years ago with what he calls system chemistry and the origins of life. And the basic premises of John Sutherland and of the people following him are that you need to have an experimental evaluation of the prebiotic system chemistry of various subsystems, and then you can use any commonality of intermediates and or byproducts to link the subsystems. Let me give you a very simple experiment, a very simple scheme of this. You can have one way of synthesizing few amino acids and and activated nucleotides, pyrimidine nucleotides, but you can use the glyceraldehyde that you have used to synthesize these amino acids also to form lipid precursors. And you can, on the other hand, use hydrogen cyanide in the presence of copper, which is not very prebiotic, I should say, but nevertheless, you can actually see the formation of glycine, glutamine, asparagine, aspartic, and then some of the intermediates or the outcomes of this experiment can be used to form other amino acids, or in combination with hydrogen cyanide, you can form activated pyrimidines. And a brilliant example of how this chemistry can work is the experiments by Matthew Porner. Actually, here it should say Matthew Porner, but my apologies, in which you see that you actually have the problem of forming the cytosine bond with the sugar, the cytidine, but in fact, you can actually make some of the compounds used from the synthesis of pentose amino oxalate. You can use them to react with the glycoaldehyde to form ribose. Then instead of going this directly from here, you can actually think of having the ribose on derived from the pentose amino oxalate, and finally you end up with activated cytidine. In other words, what you need to do is to do one experiment using synthesizing one compound that you're interested in, taking that compound, putting into another vial, into another flask, trying to synthesize a different compound and so on, and doing what one can call multi-pot prebiotic chemistry, in which you change the pH, you select the compounds and so on. And this has received a strong criticism from a number of colleagues, because people feel that we have two opposing ways, I'm not quite sure they are truly opposing, of modeling prebiotic chemistry. On the one hand, you can have the Miller-Urey one-pot laboratory simulations that can give you amino acid, hydroxy acid, purine, sugars, etc. And this is a high-energy, non-selected chemistry. And it's a one-step production. Just by one step, you produce mostly one biomolecule at a time. You have some low yields of some additional compounds, and you have also the formation of very complex organic compounds that are very difficult to analyze. On the other hand, John Sutherland approaches the system chemistry experimental approach, where you have the synthesis of precursors of ribonucleotides, amino acids, lipids, linked through cyanosulfide chemistry. This is a low energy processes. It depends on the inherently reactive favor chemistry. It's a multi-pot experimental model of different interacting chemical subsystems. It involves, unfortunately, some non-prebiotic reactants, but the same can be said of some of the experiments over here. And it requires a number of metabolic cations that could have not been present in the primitive earth like copper. So this is actually an ongoing debate that I think will eventually find a solution. But there are brilliant examples of how you can, using this system chemistry approach, you can actually produce amino acids. This figure comes from a paper of John Sutherland, and you can see a very, very complicated chemistry. It's a delight to see him explain how the whole system proceeds. But you see here the synthesis of a number of amino acids: threonine, alanine, glycine, isoleucine, arginine, serine, leucine, valine, asparagine, aspartic, glutamic, proline, and so on. Fair enough. This is giving you a number of amino acids that we are interested in. But if we compare with the meteorites, they are the best model available, the best remains available of the chemistry of the primitive solar system. What we see is actually over 90 different amino acids in different meteorites. So clearly, the abiotic synthesis of amino acids based on cyanosulfidic acid is quite successful, but it does not reflect the amino acid diversity found in carbonaceous chondrites, which we believe are the best available model of the primitive solar system chemistry. And the question is, does this imply that there were two different chemical processes taking place on the primitive Earth and on the parent body of meteorites? Once I discussed this possibility with Matthew Porner, and he says, well, perhaps this was a high energy synthesis in the parent body of the meteorites, low system chemistry working on the primitive Earth. But the question remains there. In any case, whether the Miller experiment is criticized or not, quite clearly, this is an experiment that marked the start of the experimental approach to the origins of life. And one should be actually quite happy with this. One should commemorate, one should celebrate this kind of results. Now, once, by pure chance, I remember in a bookstore, I found a novel that I felt attracted to by Harry Mulisch, a Dutch author that passed away. And the novel is called The Procedure, and it's a novel that I strongly recommend to all the younger scientists and the students attending this lecture, because it shows in a very dramatic way some of the dilemmas that as scientists, as scholars, you have to face sometimes, making a choice between your personal life, your wanting to remain in a given relationship or in a particular place, and the conditions that your work may impose upon you. And actually, in one part of the novel, one of the characters says, I was born in 1952, but my philosophical birth therefore took place in 1953, the crucial year in microbiology. It was then that Watson and Crick constructed their model of the DNA molecule, the famous double helix. In my philosophical year of birth, another remarkable experiment was conducted, to which I'm also closely connected. In the 1940s, provoked by a Soviet scientist, Oparin, there was a debate about the origin of life on Earth. His motive, of course, was, after Darwin, to eliminate God even more definitively, which is naturally the case, but still. Everybody agreed, goes on Mulisch, that a major role in the origins of life was played by 20 amino acids, the building blocks of proteins. But how had they originated? An impudent student, Stanley Miller, had the kind of brilliant thought that doesn't occur to a sensible person. Let's try it, he said. With the kind of chemical equipment that every inquisitive adolescent has in their attic, he simulated the presumed conditions of the Earth's atmosphere at the time with hydrogen, methane, ammonia, lightning, etc. And he made the sparks fly through his mixtures. And what do you think? Just after just a week, he was able to demonstrate the formation of all kinds of essential amino acids. I myself cannot complain with my aerobions. After all, even the Pope has warned mankind against me, but that pure, elegant simplicity of Miller experiments awakes in me a respectful admiration. And I think that you will agree with me that for all the limitations of the Miller experiment, that pure, elegant simplicity of Miller experiments awakes in me a respectful admiration. Thank you very much.